today I want to welcome Professor Ed Boyden, who comes to us from MIT and is one of the real leaders in optogenetics, and is going to be talking with us today about optogenetics, robotics, single-cell analysis, and other neural circuit technologies. So Ed has won a slew of awards. First, we'll start with his degrees. He did his undergraduate degrees at MIT. He has a bachelor's in physics and in electrical engineering and a master's in electrical engineering. He then went out to Stanford and got his PhD in neurosciences, correct? Just making sure that I get that right. It is neurosciences. Yeah. He has won several awards along the way. He's a NIH Innovator, one of the Innovator Awards. He has an NSF Career Award, and all of those are lovely, and we hear those from many scientists. But then he has some pretty cool ones on this list. He was an invited speaker at the World Economic Forum in Davos, Switzerland this year. Let's see, one of the others that stands, a TED invited speaker. So he's given talks at really, really cool places. He was a Discover Magazine 20 Best Scientists Under the Age of 40, which those are nice because then people know you're young, which is always cool as well. And he's really, I think we all probably know a lot about his work, but it's exciting to hear the cutting edge work because Ed does the kind of work I think that can really revolutionize neuroscience and neural engineering. So we're delighted to have you here today. Thank you for coming and talking with us.
All right, well, thank you so much for the invitation and the kind introduction as well. So I'm going to talk about today mostly unpublished new stuff that we're working on that's on the interface of trying to understand how cells in the brain work together and also how to control them in order to fix neural disorders. And so we're exploring prototype therapeutic strategies as well. And I think one of the big sort of aspects of the brain that makes it such a hard system to bioengineer is its incredible heterogeneity. Of course, all of biology is kind of dense, but the heterogeneity of the cells in the brain and their connections and how they change in disorders is truly astounding. You know, within a cubic millimeter of brain tissue, you might have 100,000 cells with 1 billion connections. And even a list of the kinds of cell and how they interact is still not clear. The other thing, of course, is that brain disorders will affect, of course, different regions, different circuits, and different cells in different ways. And being able to interface to specific biological functions in cells or specific cells in circuits is something that might be useful in expanding the kinds of capabilities that are accessible in neuroprosthetics. So what I'm going to talk about today are a couple strategies, but I want to start out with a new study that was just accepted to Nature Methods yesterday, which is how do you figure out how many kinds of cell there are? And also, if you do find cells that are interesting because either they're compromised in a disease or you find that by stimulating them or ablating them that you improve a brain disorder state, how do you figure out what the molecules in them are so that you can, for example, find better drug targets or otherwise understand the cell? So if we kind of took a step back and tried to sort of think about what cells of the brain look like in terms of their features, you might imagine some kind of high-dimensional space. This is not data, obviously, but where these axes represent different levels of gene expression or different morphological or different electrical characteristics. And I think most of us think about cells as maybe a little cluster in this space, very high expression of gene 1, low expression of gene 2, and I can't plot the other 30,000 dimensions because of the limits of my computer. But you know, this sort of cluster might define a cell type of the brain. But of course, we know that in disease, cells can change their molecular and morphological and connectivity characteristics. And so you might imagine that cells would drift along these trajectories, sort of like, you know, those little nonlinear dynamics patterns you see when people talk about chaos and the weather and so on, some of which we might be experiencing this year. And so these cells are going to be traversing these different points. Maybe this is the normal state, and then over a period of time, these cells drift into a different state. And some cells might not have any state at all. They might be continuously orbiting around as the motivational state or circadian rhythms or other things change. So one common idea is that we can try to look at cells as populations. But there are possibilities where this might not be the case. So for example, imagine these green cells are all of a given class. And suppose we took the average properties of that class. We would get this sort of little open circle there. Because that's the average of all these points, let's say. But that average cell doesn't actually correspond to a practical feasible cell of this type, because this shape is not like a blob. It's got a center that's not in the actual shape itself. And so we decided that we needed to try and figure out some technologies that would allow us to not only find and classify cell types in the brain in a high throughput, robotic, integrative fashion, but also would be easy enough to use that people could use them themselves to assess cell state, because cell types, as we've just talked about, might be drifting around all the time. So we thought about this for several years. We finally decided that there was a technology that was already out there. In fact, in various incarnations, it's been around for decades, which is patch clamping, where you take a pipette, you bring it next to a cell, and then you apply a little suction. And if you do it just right, you can get intracellular access to the cell. And now you can record it electrically. You can harvest the contents to look at the gene expression patterns, and maybe even someday soon the microRNA profiles and other programs that make the cell what it is. And of course, you can fill it with a dye so you can see its morphology and where it projects to. And so that's exemplified here. We can record, we can fill it with a dye, and we can do single cell quantitative PCR or RNA sequencing or that kind of thing. This is all, I should point out, a collaboration with Craig Forrest, a mechanical engineer at Georgia Tech. And we have a joint graduate student, Suhaza Godandaramaya, who's been working back and forth between our groups. So in vivo, patch clamping has been sort of an art form. There are maybe a couple handfuls of groups in the world that do it really well. In vitro, of course, you can patch clamp using robotic technologies, but it's been hard to generalize it to the in vivo case in the living brain, where you could actually do these kinds of observations in a disease state or in an intact circuit. So we got together and built a fairly simple robot. It's actually just one extra motor and some electronics, and I'll tell you how it works in a second. That was easy. That only took about a day or two. But then what was hard was figuring out how to actually execute this algorithm. How do we stop on this little thin lipid bilayer so that we can get a cell without injuring it? And how do we then get precision access to the inside of the cell so we can do all these things without destroying things? And so I'll tell you the specs first, which is that we can actually detect a cell in this fashion with better than 90% yield in a couple minutes. And we can do this intracellular access, which is what enables the harvesting and the morphology and all that, in another couple of minutes with, for a really good surgery, about 70% yield. For an average scenario, maybe about 50% yield. And the way it works is we're actually just using this little micropipette, this glass tube, as sort of a zero-dimensional camera. We're lowering it into the brain, and we're just continuously measuring the electrical impedance between the inside and the outside of the pipette. And that's kind of how humans do this, but humans also look for a lot of patterns in it. And so we had to figure out what would be the best algorithmic pattern detection strategy. And so it turns out that looking at the time series of the pipette impedance is better than looking at any one slot. So what the robot does is 10 times a second, it is delivering a little pulse of voltage, and then we're measuring the current that comes through. So this is the voltage clamp methodology. And then we're looking for small but monotonic increases in the electrical impedance, which would indicate the presence of something that's occluding the electrical path away from the pipette. And so this this fast but small and reliable change right here is actually a cell being detected. So as you can see, it's much smaller than what a human might be able to tell. And because we're sampling it 10 times a second, and now we're exploring whether we can go even faster, maybe 100 times a second, the robot can stop within about a micron or two of where it wants to be. And so that's why we get the extremely fast performance, the very high yields, and the pretty good success rates. This is the whole algorithm. So it took about a year for us to kind of work through all the parts. But now that we have one, it's actually quite easy to tune the algorithm. So for example, suppose that we wanted to take, and we're actually now starting to try to explore this, human brain samples that are removed after a neurosurgery event, for example. And we'd like to figure out in this specific patient, what are these cells, how do they vary from one patient to the other? Can we do personalized medicine for neurosurgery, as it were. And that's important because many neurosurgical events require follow-up that is pharmaceutical in nature. Epilepsy resections, for example, often require a little bit of, or much, depending on the patient, follow-on with anticonvulsants or anti-epileptic drugs. If we could try to classify the cells within that piece of brain tissue, maybe we could help with the post-surgical follow-up treatment in a way that would be informed by the actual cells from that person's brain. So basically what we can do is tune the algorithm. This is the detection algorithm right here. We don't have to get bogged down in all the technical mumbo-jumbo. But we're moving it at a certain point, and we're looking for a threshold. And this loop is basically the algorithm. So we can use it to hunt for cells of one kind. We can go for deeper targets or more shallow targets. And then we can form the seal onto the membrane and then open up intracellular access. We derived this all for the mouse, the anesthetized but live mouse cortex. So that you can see here the up and down states that are characteristic of the mouse cortex. But it works pretty well in other regions too. So it has equal yield in the hippocampus, and we've been able to go several millimeters deep into the brain. So we think that this might, with some additional work, be a generalizable strategy for doing high-throughput single-cell integrative analysis in living brain. And this is a very dense slide, but basically on the y-axis are various measures of the quality of the electrical recording, and the take-home message is that they're all pretty good. The grayish symbols are the manual expert single-cell patch clamp, and then the others are the automated. And what you can see is they're all kind of similar between human and machine for a very, very skilled human who's been practicing for years. And also the quality is independent of depth up to about 2 millimeters or so, which is the deepest we've gone with quantitative assessment of the quality. So to summarize this part of the talk, we really want to do integrative analysis of cells. Can we go from the program that makes a cell what it is to what it looks like from a connectivity and morphological standpoint, and to also what it looks like electrically in a given behavior or a disease state, but to make it so that people can kind of build themselves, because these robots are fairly straightforward to use, to characterize cell states in a given disease context in an animal model or in a human. And we've just started a small company to commercialize these robots. I'm not an owner or an equity partner in the country, as my anti-disclaimer. But we can collaborate with the company as a result, and so we're now working on a variety of things, including the ability to scale it up. So these are all just very realistic CAD diagrams. These are not actual devices. I wish they were. But we're now working on ways to try to scale this up to dozens and maybe even hundreds eventually of individually actuatable micropipettes. And to do that, we have to do a number of things. First of all, we have to find a way to couple lots of cheap actuators that are still micron precise to these little glass pipettes. And to do that, what we've come up with are basically fused silica flexible glass linkages so that the glass can convey force from these motors down to this little area where the action is at. The other thing is to automate the fabrication of the pipettes. And Craig's group at Georgia Tech has had a little paper showing how they can make a robot that can actually make lots and lots of these pipettes. And now we're working on a second generation of them, of that as well. The other thing we have to do is to figure out how to make this many amplifiers, because often in neurophysiology, the amplifiers are expensive. And so we're collaborating with Eugenio Colorcello at Purdue to figure out if there's a good way to do that, since he has a lot of experience in patch clamp chip things. So to summarize part one of the talk, we're really excited about this idea of making a tool that democratizes the ability to assess cell states in a rapid, high-throughput fashion. Version 1 is pretty much done, and if anybody wants the manuscript, it's going to be in final form Saturday morning as soon as I get a chance to do the final rewrites. And if you're interested, we're always looking for beta testers. People are trying to figure out all sorts of applications for this. Can you do it in an image-guided context? So far, we're just using the impedance as the detector. Can it be used, what are the variables that have to be incorporated to do this on biopsies? Right now, this is all in live mouse, which I would assume is harder than a biopsy, but we don't know for sure. And so we're eager to try to find applications of these kinds of integrative tools in a variety of contexts. Now, part of why we started this was also motivated by the next part of the talk, which was over the last several years, a lot of what we've been doing has been in the space of trying to figure out, can we turn on and off different kinds of cell in the brain? So for example, excitatory neurons or parvalbumin positive inhibitory neurons or other cell types, can we turn them on or off, which both allows us to assess their causal role in the network in which they're embedded. And also maybe if there's a cell that's underperforming or a cell that is lost, can we alter the dynamics of the circuit in order to augment the computations being performed? And in part, we started doing the single-cell robotics in part because of this question of, we didn't really know if we had one cell type or not. Maybe we're actuating or turning on or off a cell type, but it's actually 5 cell types. Or the cells change state and cells merge or change phenotype. And this is something that many groups are now showing, that during disease states, cell types kind of collapse in phenotype. Some of the diversity seems to go down. So what we've been doing, and this slide summarizes most of the published stuff that we and several other groups that I'll talk about in a second have done over the years, is to find molecules that when we express in cells allow us to turn them on or off with light. And these molecules, this is a diagram of a molecule sitting in the plasma membrane of a neuron. These molecules are all 7 transmembrane proteins from plants, microbes, fungus, a species, and so on, which have the interesting property that when light hits them, they will pump or otherwise translocate charged cations or anions across the lipid bilayer. So for example, there are light-driven outward proton pumps and light-driven inward chloride pumps, archirhodopsins and halorhodopsins respectively, that when you put them into a cell will result in the cell, of course, becoming hyperpolarized. And then we've also worked on light-driven inward cation channels, channel rhodopsins, that when hit by light will open up a pore and let cations into the cell. And these little diagrams just summarize what happens in neurons when you do this. The channel rhodopsins are the ones we worked on first, and I'll tell you about those first. When you put those into cells, then blue light can cause them to fire action potentials, was what we originally published. For halorhodopsins and archrhodopsins, you put them into cells, and you can then quiet down the neurons. Halorhodopsins turn out to not, in their natural form, to be particularly high performance. And so a variety of strategies have been adopted to overcome them, which I'll talk about in a second, including genomic mining of more molecules, such as the archrhodopsins. So here you can see a raster plot where the spikes are neurons firing away in the awake mouse cortex. And then we turn on the light, the hyperpolarization begins, and the spikes stop. We turn the light off, and the spikes begin again. So let's delve deeper into what all these little molecules do. The first molecule that kind of got the attention of us was characterized by Nagel, Hegemann, and Bomberg in 2003. And Carl Deisroth and I, both at Stanford at the time, wrote to Georg Nagel and said, hey, this looks interesting. Maybe we could use it to turn neurons on. And so this was the channelrhodopsin 2, was the first one that we worked with. And it's from green algae, where it's found in a little eye spot in the algae, which allows it to navigate towards light. So there's a little eye spot here, and then the flagella are steered by this eye spot. And the molecule looks something like this. There's a vitamin A derivative, all-trans-retinal, within it, which is nice because in mammals, this is ubiquitous, and therefore, for the most part, you don't need to add any supplemental chemicals for use in the living nervous system or body. In other organisms, like Drosophila and C. elegans, people have been able to feed them the retinal. As we already talked about, when you illuminate with light, you let in positively charged ions. And luckily, several bits of serendipity aligned. First of all, it was nicely encoded by a small enough gene that we could put it in using the small viruses that we have available to us. And also, although it localizes to an eye spot in the original organism, for some reason it ends up on the plasma membrane of neurons, and also it has the right speed and amplitude to mediate action potentials. So being lucky is important, I guess. And I always tell the students in the group that this is a good example of why they should try to be lucky. The other thing, of course, you have to do is to get light into the brain. And many, many groups and companies now sell or have made things like this, where you just take an optical fiber and put it into the brain. Originally, the strategy from the Aravinus et al. Paper was to put an optical fiber into the brain that was connected to something outside, but breakage of the optical fiber has led many groups to adopt a second strategy, which is to have a chronically implanted small optical fiber. Imagine the skull right here. So this fiber is in the brain pointing at a site, and this connector sticks out, and then you can plug in a, say, optical fiber attached to a laser or a light-emitting diode or whatever else so that you can illuminate into the brain. Now, as we talked about, we don't have a list of the cell types of the brain, and it's not even clear whether cell types are static entities. They probably aren't. But nevertheless, we need some way of targeting the cell types as well as we can describe them now with these tools in order to turn them on and off. Because, of course, if we don't target single kinds of cell, then it doesn't really offer any advantage. So one strategy is that you can find viruses that have certain serotypes or coat proteins that allow them to bind to and be taken up by some cells and not others. And this has been pursued by some folks, but it's kind of hit or miss, right? You have to just try things out. And so another popular strategy has been to tune the DNA regulatory elements or promoters that are in the virus so that only cells that drive that promoter expression will actually express the gene of interest, say the channelrhodopsin 2. And probably about a dozen promoters have been published with reasonably good specificities that you could hit dopamine neurons, inhibitory neurons, excitatory neurons, and so on and so forth. But a lot of, at least in the basic science side of things, studies have taken advantage of all of the intersectional genetic strategies that are out there. So there's many classical organisms in neuroscience and biology in general where you have a driver line and a reporter line. And the driver line basically expresses a molecule that's a driver molecule in some cell type, and then the reporter line expresses the reporter, a fluorophore, an optogenetic reagent, such that the driver will bind to that thing and turn it on. So, for example, there are over 200 now lines of mice that express Cre recombinase under a specific promoter, such as the promoters that enable dopamine or serotonin or other cell type markers to be made. And what you can do is inject a virus that acts like the reporter, which has a ubiquitous promoter. And what happens is you just have a cassette that is inactive, but its expression is activated by this driver. So in this case, Cre recombinase is a molecule that binds to these little triangle pieces of DNA. They're not actually triangles, that's just a symbol. And what happens is, in this case, this reversed molecule we flipped around to read in the forward direction. And so this strategy was published by 3 groups around the same time to actually enable these driver mice to be used with these reporter viruses. And I'll tell you in a second about a paper that's going to come out pretty soon about how you can do this all purely with transgenics. So let me give an example of the kinds of things you can do with this. This is some data that was from a collaboration led by Christopher Fiorillo. That's from a paper that should be coming out pretty soon, which is to use the dopamine Cre mice, which use that example of intersectional genetic targeting we just talked about, and then that reversed molecule trick, so that only this virus might be taken up by many cells, but only will the expression be activated in the dopamine transporter expressing neurons. And the basic idea was to try to figure out, how brief an activation of the dopamine neurons do you need in order to get some kind of behavioral effect. Dopamine neurons, of course, are implicated in all sorts of really powerful and interesting things in normal and abnormal behavior, and also are clinically extremely important. So the assay that Kyungman Kim, the graduate student who was working between our groups, did was to build a little box, and every time the mouse went here, it would get a pulse of light, and every time the mouse went here, it wouldn't. So this is that mouse, and it's getting a 200 millisecond pulse of light every time it goes to this point. It's poking its nose into a little sensor. And as you can see, it'll go here over and over and over again, and it won't go to the other one. So this allows us to do all sorts of interesting things, including what you might think of as maybe screening for targets in the brain that might be involved with addiction. And so to generalize this idea, we started designing, this is work that Jake Bernstein did in the group, strategies for illuminating independently many sites in the brain. So here we have about a dozen optical fibers, a little bit more than that, each coupled to its own light-emitting diode. And this is all assemblable in a quasi-manual way. Each LED is independently controlled by electronics, and these can bilaterally tile, for example, the mouse hippocampus, that you can go through the brain and look for parts of the brain that are involved with memory or epilepsy or other things like that. And this led to this idea that you might be able to do high-throughput circuit screening to go through the brain and bit by bit activate something to figure out which part is most important. Then, of course, you have to either use that for some clinical or scientific use. Suppose that a target is found. Maybe that target now is a better place to put an electrode, or that target could be directly optogenetically actuated. Or clinically, one could also imagine using the robot to come in and then harvest the molecules and the gene expression patterns in those cells and convert those circuit-level targets into drug targets. And so this idea of high-throughput circuit screening is an interesting one, because you might be able to rapidly go through the brain, figure out which cells and regions are most important for a clinical benefit in an animal model, and then find drugs that might be selective for those cells and not others. Few molecules are actually druggable targets, maybe 10% or less. But with the gene expression profiling, you get very high-dimensional profiles of cells. And so we have a hope that even though this is a high-risk kind of environment, the ability to get very high-dimensional signatures of individual cells densely arrayed in a single brain region might enable many new molecular handles to be acquired for cells. So in this vein, to do this kind of screening, Christian Wentz, who was a master's student, developed a wireless optogenic device, which he also spun out into a company, which again I don't have any ownership interest in. But this is a wireless device here. There's no battery, and there's no wire coming in, and there's no optical fiber coming in from the outside. Imagine the LED plus or minus the fiber on the bottom of the circuit board, and then there's a little antenna here which can pick up magnetically delivered, inductively coupled power. And then there's a little bit of supercapacitor circuitry, which I won't go into because it's all published, which allows the power to be intelligently managed. So here's this mouse. It's on a magnetic coil. There's the receiver. And this is good enough that we can actually get several watts of power continuously delivered to this headpiece on a mouse as it walks around. Here's another arena that we're constructing. And you can control maybe 83 mice from your laptop. You just plug in this little dongle thing. And it works. So this is movement on the y-axis, and here are a couple control conditions. And then when the LED is on and the power is on, then the animal will start to move. Because in this case, the LED is implanted over the motor cortex, and this is a mouse where the motor cortex has been made transgenically sensitive to light. It's one of Guoping Feng's transgenic mice that have ubiquitous expression, say, across layer 5 of cortex. So you can check this out at Best Buy as the next generation of wireless optical mice. which now that we're the iPad generation, nobody will get that joke in 10 years. So, we're working a lot on expanding the molecular repertoire of what can be done with these optogenetic tools. And I'll tell you about two short unpublished stories now, where in the channelrhodopsin space, we've been working on mining genomes of hundreds of algae that the 1,000 plant project, of which we are a part, led by Gain Wong at the University of Alberta, have been sequencing. And so these are all algae that are driven by the Melkonian group at the University of Cologne. And we're finding all sorts of molecules with very interesting properties. So I want to tell you about a couple of them now. One thing that has become clear is that in many cases, these molecules can take some time to express. Some of it obviously depends not only on the molecule type, but also the method of gene delivery. But in many cases, it can take some time to express. And for example, many groups that even just electroporate the genes in, which is about as direct a way of getting in as you can, find that it takes a month or more to get good labeling of cells and their processes with a channelrhodopsin 2 molecule, for example. So one of the molecules we've found, which we've nicknamed CHR87, seems to express very, very quickly in a side-by-side comparison with the same methods of gene delivery used for the CHR2 molecule. So in this case, you can see it expressing all over these cells. And this is all down the axons and dendrites of neurons 1 week after birth, whereas usually groups are waiting 3 to 4 weeks or even longer to get this kind of expression. So one possibility is that maybe this molecule might be a better translational candidate. It might express faster. It might fold better as proteins go. We're still gearing up to investigate the phenotype of why this is so. We have a grant starting pretty soon, which is all about trying to understand the principles of sort of heterologous expression of these plant and other proteins in a safe and powerful fashion. But nevertheless, at a phenomenological standpoint, it can express quickly, and that's good. I'll tell you another story, which is about color. So this is photocurrent normalized to the peak on the y-axis and color on the x-axis. And the original channelrhodopsin is this blue curve here. It peaks in the blue, which is nice. And there's been a lot of attempts to try to get multiple colors. You can do all sorts of things with multiple colors. Red light, of course, can go much farther into the body than blue and green light. So this is a pretty powerful green laser pointer. I'm shining it through my finger, and you don't see anything. If you take a flashlight, though, which is much, much weaker in terms of irradiance, you can shine it through your hand, right? And so, and what comes out is the reddish light. So there have been lots of attempts. These two papers were published by the Hegemon and Dijsselroth groups over the last couple of years to try and find redder channelrhodopsins, and these are peaking in the green. By mining this database, this is work led by Nathan Klepetke in our group, we found this red channelrhodopsin that we nicknamed Crimson. And as you can see, 633 nanometer light, which is like a red laser pointer, will give you appreciable depolarizations, whereas with the older molecules, it's all but negligible at that level of power. We've also been able to find bluer channelrhodopsins that are out in the purple. And so you might imagine that maybe we can use the red ones to go deeper into tissue. And by using the bluer and redder ones together, maybe we can do two-color activation. We can control two separate populations in the same optical field of view, as it were, without having crosstalk. I'll show you some demos in a second. But I just wanted to mention this is now the maximum current. And we've looked at a bunch of molecules like this. You can see most of them are blue and green, like the original ones that we all published. A few of them are purple, and we've only found one that's red so far. So it might be that much in the same way that green fluorescent protein from jellyfish can't be made red by mutating it. You have to go to corals to get red fluorescent proteins. It could be that there are distinct classes of color mutants that exist out in the wild, and they're very hard to transmute one to the other purely through mutagenic means, although that remains to be seen. The photocurrents are also pretty good with the new ones. We have one new one, Chronos, that has particularly high photocurrents, and we're still exploring its properties. It's kind of a weird molecule, so I don't have anything more to show you today on that. But let's look at the red one, Crimson. So this is Crimson expressing in the for electroporation. I should point out that Martha Constantine-Patton's group at MIT did all the electropораtions and the physiology. And what you can see is that modest red light can drive action potentials in these visual cortex neurons. In this case, it's a brain slice. Blue light, on the other hand, barely causes anything at all, and certainly does not cause action potentials. And so that means that there's a window now where we can use blue light to drive stuff with the bluer channelrhodopsins, and red light can be used to drive crimson expressing neurons. And so we could do things like activate a modulator pathway and activate a excitatory pathway to see how they interact, or to see how the timing of two independent pathways interacts at a cellular and biophysical level. So From a prosthetic standpoint, this also might be of use, but the examples are a bit more complicated there. But in basic science, there's all sorts of questions about how two pathways interact that remain to be answered. So I want to switch gears to silencers now. Silencers, of course, come in a couple different classes. We already talked about light-directed inward chloride pumps. And I wanted to talk a little bit about the path, because it illustrates some important principles about these reagents and where they might be of use, but also where they might need improvement. So what kicked off the hunt for the light-driven chloride pumps was this paper that came out from a group from Japan in the late 90s. And the halorhodopsins come from organisms that live in very high salt concentration environments. So this orange bar is where the brain is at. And you can see this organism has its peak photocurrents of its halorhodopsins around 5 molar chloride, which I think even in neuropathology never happens in the brain. But one of these molecules was different from the rest. And for whatever bizarre evolutionary reason, it had its peak splat in the middle of the brain chloride concentration range. And so that definitely caught our attention. So our group and also the Dijsselroth group, working independently, both published papers around the same time showing that when you put this molecule into neurons, indeed you can get hyperpolarizing currents, although they're modest. They're about 10-fold lower than the currents I just showed you on that bar graph for the channelrhodopsins. But nevertheless, it was a good proof of concept showing that you could actually quiet down cells in the brain. Now, the currents are low, and so it wasn't really till last year, 4 years after the publication, that a mammalian behavior was perturbed using the original N-phoronous halorhodopsin. It works quite well in C. elegans and other in invertebrates, but for some reason in mammalian cells, it doesn't necessarily express at very high levels for various reasons that are still not completely clear, but which can be ameliorated in part by boosting the trafficking, as Guoping Feng and Carl Dijsselroth groups have explored. Nevertheless, this one paper from Akihiro Yamanaka's group did show that the original N-phoronous halorhodopsin, when expressed in the hypercretin or orexin cells, which are, of course, for the cells that are compromised in narcolepsy, can be safely expressed without any agglomerations or problems. And then when you shine yellow light on these cells, the animals will fall asleep. So this is probability of wakefulness, and this is time in seconds. And you can see when the light goes on, the mouse just go into slow wave sleep. And then you turn the light off, and they wake back up. But they're still groggy. Not unlike an audience at 9:04 a.m. when the talk began at 8:30. So this is interesting because, of course, the evidence was strongly pointed towards the idea that narcolepsy was mediated by the loss of these, but it didn't prove that these cells acutely were involved with the falling asleep transition. Maybe there was some plasticity elsewhere in the brain that was happening. And so it's interesting because there actually is a 20 to 30 second lag until the mice fall asleep, which raises the question of how is this effect propagating throughout the nervous system? Because, of course, sleep is a property that ends up involving a big chunk of the brain. So we wanted to improve the photocurrents. And so Brian Shao and Shira Han, who are now faculty at UPenn and BU, got together, and we started screening through molecules just from all sorts of candidate organisms that might have good silencers. And so we found one of these molecules that had particularly good membrane expression, and it was green light driven, which had photocurrents that were about an order of magnitude bigger than the original N-phoronous halorhodopsin, both at low light powers and at high light powers. And so this is what yielded that first 100% shutoff in the awake behaving mouse that I showed you earlier. So this molecule we nicknamed ArchT because it came from an archaeorhodopsin class molecule. And interestingly, when we eliminated all of the usual suspect ions, it still worked. So this represented light-driven outward proton pumps. And protons are usually something that in neuroscience we're trained to fear. So we wanted to figure out, what does this do in neurons? So we did a lot of phenomenology studies, both in primates and in mice, and showed that before and after minutes of silencing, there wasn't any change in spike firing rate or spike shape and so on. But we also wanted to figure out biophysically what was going on. So we got some advice from Roger Chen's group on measuring, John Lin and Roger Chen's group, on measuring pH changes with pH dyes. And we found that indeed there was a slight alkalinization of the cell contents, but it was pretty small, 0.1 to 0.15. And actually the reason that John Lin and Roger Chen were measuring pH was that they were measuring the pH with the channelrhodopsins and found that they had even bigger proton fluxes and bigger pH changes. Channelrhodopsin 2 actually has a proton conductance that's 1 million times higher than its sodium conductance. Which might lead to interesting properties, actually. Maybe you could even put them into mitochondria or chloroplasts and boost photosynthesis or make photosynthetic animals or something like that. But the channels have a much bigger conductances than pumps, right? Pumps are moving 1 ion per cycle or maybe a little bit more if you see some of the branch photocycles. Channels, on the other hand, can translocate hundreds or thousands or more ions per photon. So we've distributed these ArchT class molecules to hundreds of groups, and I'll just tell you one short story of a paper that's under review, which was a collaboration with Kai Goosen's group, which was how does serotonin have its effects on psychiatrically relevant conditions in the brain. So what we did was use that same trick, the serotonin Cree mice, serotonin transporter Cree mice, and then we put in ArchT now, so we could label those neurons. And then by recording the serotonin neurons, we could see that the light could do a temporally precise silencing. This is actually using an earlier version of the patch machine that was not automated at the time. And this is the population data showing we can turn off serotonin neurons in a temporally precise fashion. Now, the question we wanted to answer was, how does stress have its impact on the brain? And that's a complicated problem. But serotonin has been implicated both in positive and negative fashions with the effects of stress on the brain. Obviously, SSRIs and antidepressants have as one of their features serotonin-acting effects. But we wanted to try to see if we could parse some of this out using temporally precise perturbation of the serotonergic neurons. So what you see here is a paradigm where mice are experiencing 2 days of a widely used stress paradigm where they have an hour in a small container, so it's not painful, but it's a mild stressor. And then on the third day, they undergo Pavlovian conditioning, which is in its classical form, a tone followed by a mild foot shock. And on day 4, you play the tone, and you see, do they still fear the tone or not? And so if they do fear the tone, they're going to freeze, which is indicative of the memory that the tone implicates, the prediction of a shock. So we tried a bunch of things. One thing we did was to try to turn off the serotonin neurons. A lot of work on these states is focused on boosting serotonin. But we also want to figure out what serotonin could do in an aversive style, which has also been studied quite a bit, using microdialysis, for example, showing that serotonin levels will go up when aversive stimuli are presented. So we decided, driven by that idea, to turn off the serotonergic neurons during the Pavlovian conditioning. And we decided to try different parts of the Pavlovian conditioning day. This is day 3, like during the shocks. So this graph shows what we found. This is all work that was led by Mike Barada, who's now working at the University of Colorado. So the blue bar right here is normal Pavlovian fear conditioning. And this blue bar here is Pavlovian fear conditioning after stress. So these animals exhibit more fear than the animals that had fear conditioning alone. And this is interesting because clinically, people have also noted things like people who returned from a battle zone when they had a stressful life at home are more likely to get PTSD than people who didn't have stress at home. And so there might be some analogs between this kind of irrational boost in fear and the kinds of things that occur in humans as well. Now, the red bar here is what happens if we turn off the serotonin neurons during the shock of just normal Pavlovian fear conditioning. And as you can see, nothing happens. But this red bar here is when the animal had stress before. And what you can see is that when we silence during the Pavlovian conditioning, during the shocks, we block the effects of stress. And so this tells us 2 things. First of all, that serotonin can act over very short timescales to mediate psychiatrically relevant changes. And a lot of the SSRI literature has pointed us to thinking about longer timescales. But until now, it's been hard to probe the fine timescales. The second thing is that this tells us that reducing serotonin during these brief stimuli can also be important. And that's important as well because of the ongoing debates about serotonin increases and decreases. If we silence during the tones, nothing happens. So these animals did not have the stress effects of fear abolished. They looked rather like the ones that had stress before fear and were not treated. So what it suggests is indeed that aversive stimuli seem to be engaging the serotonin system or being boosted by the serotonin system. This just shows the third condition of tones with shocks. I didn't explain the paradigm because of in the detailed fashion, but if we silence during tones paired with shocks, then that also blocks the effect. So it isn't changing the learning, it's really gating the impact of the aversive stimuli, as it were. It's not gating the associative event of the conjunction of the two, but it's really modulating the impact of the aversive stimuli itself. So anyway, that's an example of the kinds of things that can be done that are difficult using purely older technologies. I want to share some data which is kindly shared by Honghui Zheng, who has a paper that's going to come out soon, which is how you can do this without the need to keep injecting viruses, at least for research purposes. And she and her team at the Allen Institute for Brain Science have made transgenic mice that express chamaeropsins, archaeodopsins, and boosted halorodopsins, like the ones that I mentioned earlier, in transgenic form. So you take this mouse, and it has basically a version of that Cree-dependent cassette in the genome now of the mouse. You can just breed it with the driver line, and the offspring will express these molecules in the cells that are expressing the Cree recombinase. And so you can do things like neural silencing. This is from the AI35 line, the ArchT mouse, where you can get silencing currents both at low and high light powers. This is using the AI39 mice, which is the enhanced halo-rhodopsin, which still works, although you get lower currents. And so these mice are all available from the Jackson laboratory, and you can just order them off the internet. This shows some data from that paper where you can activate neurons using the channel-rhodopsin line, and you can quiet them down using the ArchT line in the awake behaving mouse. And so many groups are using these mice for various things. We've been working a lot to improve these molecules. I'll just mention one slide very briefly, which is that we found a more light-sensitive version of ArchT that we named ArchT. It's 3.5 times more light-sensitive. And so in-house, that's one of the molecules that we use most routinely. But we have some new molecules that we think are really interesting, and I'll tell you about that next. A couple years ago, we published the first demonstration of multicolor silencing, which was, again, this is current now versus color. And the red curve is the original Enferonis halo-rhodopsin spectrum. And then this blue one here is this molecule that we found, rather I should say it was discovered by a group several years ago, Leonid Brown's group, from a fungus of all things, Leptosphaera maculans. And so we tried it out in neurons, and it worked. And we nicknamed it Mac, since Leptosphaera maculans opsin took too long to say. If we work with a mutant of it that's better, we'll call it Big Mac. But anyway, so this is if you have a red light opsin expressing neuron, the halo-rhodopsin expressing neuron, then red light will quiet it down, but blue light won't have much of an effect. For the Mac expressing neuron, blue light will quiet it down, but red light won't have much of an effect. And so this is a demonstration that you can also do, in principle, zero crosstalk two-color silencing, although this was only done in culture. And so in vivo, or in a slice where the light's going to fall off, it might be a little bit harder to bridge this gap because of the large spectral overlap. Whereas we had purple versus red before, this is kind of a green versus, or green versus red, which is a lot closer. So we wanted to push this even further. And so work that Amy Chuang is doing is trying to find even redder halo-rhodopsins and bluer silencers. And as I mentioned earlier, we want to do two-color because we can parse out how two different pathways interact. But we also want to do two-color because red light can go further into the body. So this just shows absorption by hemoglobin versus color. And you can see that yellow, green, and blue are all very much absorbed by hemoglobin. Red light, on the other hand, is near the hemoglobin trough, and the absorption is down by about a factor of 100. And similar trends hold for other components of brain, like lipid and so on, although maybe to a slightly lesser extent. So we found a molecule that does have a red-shifted action spectrum. This is the original Enferonis halo-rhodopsin in blue here now. And this red one is this halo-rhodopsin 57. It might look like it's not that shifted. But if you're out on the red, where the slope is very steep, then a small shift can lead to a large boost in current, about threefold, as you can see here in this normalized spectrum. And it equates to maybe about 2- or 3-fold in raw current terms as well, over, let's say, the ENPHR3 that we just talked about in the transgenic slide. And also expresses pretty well in the mouse brain. So we're getting good expression. So this allows people to then do red light silencing. And we're trying to figure out how big a volume can be silenced. Could it be, things on the size of cubic centimeters or bigger? So as I mentioned, there's a lot of non-circuit applications that I think have not been explored very much at all, and might be even capable of helping out entirely other fields. One thing is that so far we mostly treated all of these different molecules as if they're just controlling voltage. But of course, voltage in biology has to be correlated with some actual charge of some kind. And most of that charge, of course, is ionic in nature. And so these are the ionic conductances of channel-rhodopsin 2 for protons, sodium, and potassium. What you see is that, as I mentioned, the proton conductance is 1,000,000 times higher than the sodium conductance, which is a little bit higher than the potassium conductance. And so one can imagine that you could put this into lysosomes or synaptic vesicles or mitochondria and use this as a light-gated sort of short circuit. You could short out the mitochondria for a few seconds and see if that changes a high-speed process that occurs at a specific synapse. Or protein degradation, neurotransmitter loading, all sorts of processes are proton flux dependent. And by using this as a proton conductance, new capabilities might be possible. Another paper from the Bomber group showed that you could also make channel-rhodopsins that have a higher calcium conductance. They found one that had a much higher calcium conductance, several-fold higher. And you could use this as a way of delivering calcium to defined parts of a cell. Although there are also natural molecules like melanopsins, which are in our retinal ganglion cells that are GQ-coupled GPCRs, and those can be used as well. And of course, the proton pumps, as I mentioned, can be used to create gradients, and maybe that would be of use for powering up things. On that note, a medical student, Albert Kwan, did spend a little bit of time making a mitochondrion-targeted light-driven proton pump. So this is a mitochondrial stain, and here's the pump, and you can see they overlay pretty much perfectly. But then he went back to medical school, and I don't know anything more about mitochondria. So if anybody wants this cell line, we're happy to share it. Maybe this is a photosynthetic mammalian cell. So to summarize this part of the talk, we've given out reagents, and now that we've also been partnering with many viral and transgenic distributors, probably to more like 700 or 800 labs by now. This slide is out of date. But we also like to host visitors. 1 or 2 people per week typically are stopping by to see devices or to talk about molecules or to see procedures or to see new crazy inventions like the auto patch robot or other things we're working on that I don't have time to talk about. And so we're always happy to teach, because I think that's a big part of technology is teaching. But also, we get to learn a lot about the problems that people have, and that can inspire new kinds of invention as well, such as the big effort we're now putting into single-cell resolution, high-throughput, scalable analysis. So for the final part of the talk, I just want to talk about some different ways that this could be potentially scaled up to looking at entire neural circuits. And one way that it's almost kind of trivial, we published a couple of papers on over the last couple of years, is just to take awake mouse fMRI, which is commonly done, although you have to have a scanner, and awake mouse optogenetics, which is also commonly done, and to combine the two. So we teamed up with a whole bunch of groups, Chris Moore, Ann Grabiel, Randy Butner, Nancy Capel, and built a little MRI-compatible optogenetic rig, which allows you to optically drive or silence specific cell types and then to map the brain-wide impact, which you can see here, we're using a 9.4 Tesla scanner. You can get pretty good images, maybe 50 or 100 micron resolution, depending on how big a volume you want to scan. Of course, the bigger the volume, there's a trade-off where your resolution will inevitably have a little bit of sacrifice if you want to scan. But we wrote some software also allows you to photoactivate an area and then to pull out the set of connected regions. So here, for example, this is the median of 5 mice, and then this is a 3D grid that corresponds to the rectilinear coordinates of the brain. So we're photoactivating somatosensory cortex S1, and what we see are clusters within the striatum, and then M1 and contralateral S1 and other areas that you'd expect to see. And so this is pretty straightforward to use. We can use it as a way of just mapping circuits to figure out where we could park electrodes so that we can try to drive cells in a region and then to check out what's happening in other regions. Or you could try to use this to see how drugs modulate connectivity of regions. So this is an example where we just did the experiment that I showed you in the previous slide, photoactivate S1, map the rest of the circuit, and you see the big light ups in the motor cortex and the sensory cortex and the striatum. And then we anesthetized the mouse, just giving isoflurane. And what we saw was that the activity changed. Now, what's plotted here is actually the correlation of activity in the two regions. So they're fluctuating in synchrony as we drive the pulses of light. And the red means there's a lot of coordination between the activity patterns, the bold signal or the fMRI signal. And a cooler color, like this green line, means there's not so much correlation. We're driving it and stuff is fluctuating, but not in a very temporally coordinated way. When we add anesthesia, then the correlations drop. But what you can see is that the striatum just sort of drops out of the circuit. So subcortical-cortical connectivity seems to be one of the things that's most disrupted when anesthesia is added. And so this might be a great way of building translational bridges between causal animal data and human self-report behavior and neurology and psychiatry. And so we're very eager to explore this. Right now we just use it a lot just for sort of checking where we need to go with our probes next. But I think it can be of use as a way to try to connect the dots between the incredibly powerful human and non-invasive imaging and the ability to kind of do systematic maps of circuits in animal brain. That said, we were inspired to try to do 3D neural recording as well. And so we do have a project to try to do with Cliff Fonstad's group that's spearheaded by Jörg Schulven and Anthony Zorzos, to build 3D recording arrays. So we're doing just classical silicon micromachining to put together probes where we could treat them sort of in a Michigan probe style, putting electrodes that go up and down these. And so that's still a work in progress. So I don't have any data to show you. But what I do have to show you is that we've been building 3D light probes, and those are now to the point of working. So let me explain how this works. So that 3D probe was a 2D grid of one-dimensional fingers. Let's zoom in on one of those one-dimensional fingers now. And what we're doing here is we want to emit light at many points along this probe. So we tried for actually 2 years to put light sources along the probe, but you get a lot of heating. And so we decided to put that on hold for a little while until light sources got better. And so we decided to build micro waveguide arrays. So we have waveguides that can guide light into the brain and there's a little mirror that can then deflect the light out at a 90-degree angle. And so we published this prototype about a year and a half ago, I guess. So you can have many independent channels. You can have independent lasers or whatever at the top, and they'll guide light down to different depths where they will then be reflected out to the side. So that's what these waveguide arrays look like in cross-section. We have a little bit of metal which acts as the mirror because, of course, this is a very tight turn radius, much too tight a turn radius to be good for just totally relying on the internal reflection properties. And this is what it looks like in EM cross-section. And the probes work. So this is what the probes look like when you just look at them in a regular old bright field or stereo microscope. And this is what they look like when they're on. So here we are. We have 3 lasers. We're driving light into these different waveguides, and then the mirror makes the light turn 90 degrees when they're at the desired depth. So we're still working on the electrode part of this because we wanted to find ways to put optical drive and electro recording in the same device. But we do have the 3D waveguide arrays working. And so I just wanted to show you what those look like. So this is each of these fingers is like that thing I just showed you. And now we've built them in 2D comb-like arrays and then assembled them into a 3D array by building some holders out of silicon. And that's what you can see here. The little shiny and dark alternating parts are you can kind of see where the output of those waveguides is because of the difference in material and also maybe a hint of the mirror. For testing, all we do is we take this 3D array and then hook it up to a flexible fiber bundle and then some optics that then connect it to basically a projector. So this is just a micro mirror array like you find in high-end projectors. And we take out the bulb and we put in a laser so that the mirrors can bounce light into these individual pathways. And so if you think about it, let's say a projector is a 1024 by 768 projector. In theory, that gives you something like 700, 800,000 pixels. In practice, though, what we're seeing is that we have some laser speckle when we do this off these mirrors. And so what we're actually doing is averaging several mirrors into one lens to go into a single bundle. But still, that gives you 50,000 channels of optical activation, which is a lot. So we're just playing around with these things now. So this, for example, we're activating the 111 coordinate of the 3D array. This is a prototype with only 240 waveguides. But the idea is, can we really control the brain at a resolution that allows us to kind of talk to it in a way that might be a little bit more realistic than just a whole region being turned on or off at once, which is sort of an artificial way to use light to turn on or off a cell class, I think. And here we are turning on the 531 coordinate, for example. We're working on ways to implant these things. This is a micro CT scan of a mouse. So this is the ear, and this is the skull. And you can see the array kind of there. And even figuring out how to do the histology is actually turning out to be tricky because putting in the array and then removing it, sometimes you can, with this many probes, warp a little bit of the structure of fixed tissue. So what we're doing is taking the fixed brain and scanning it in a CT scanner to try to figure out where the tines of the probes are with respect to the ventricles and the different parts of the brain. So I think I'm going to end on this kind of note. Maybe I just want to have one last slide to talk a little bit about what kinds of potential clinical paths might there be. And obviously, you need a couple things for this to work. You need an invasive implant of some kind. And of course, it's not news here, of course, where a lot of the pioneering work has been done. That invasive electrical stimulation, of course, is something that is growing exponentially in use in the clinic. Gene therapy, of course, is needed to get the genes in. But even that's seeing a sort of resurgence in interest, especially with the adeno-associated virus, not to be confused with the adenovirus, which has been used in many hundreds of patients in clinical trials without any serious adverse events to my knowledge. So the elephant in the room, of course, is these are molecules from algae and bacteria and funguses. And so would the body detect them as foreign? Will there be a reaction to them? So we collaborated with Bob Desimone's group and Anne Grabiel's group to try out some primate preclinical work to see what would happen. So we put the molecules into the brain and photoactivated them and saw that indeed there wasn't a decline in function over time, although to be upfront, these are very occasional tests. And then in a small number of animals, we did histology to see whether there were any evidences of immune cell infiltration or neurite derangement or gliosis and so on. And it's very early days. This is only a handful of animals, but it does seem that over a period of at least many months, up to about a year, which is as far as we've looked, that you don't see overt evidence of the kinds of damage that would ring alarm bells. So it's very early days, but many other groups are now doing primate tests as well. And so far, things are looking positive. So if the science continues to be borne out, there might be some hope for use of these things directly in a clinical setting. So with that, I just want to acknowledge, I think I mentioned many of the people, but I always forget a few as I go quickly through things. So I wanted to mention, so Brian Allen and a very talented undergraduate, Rachel Bandler, have been working at the micro-CT probe localization. Jake Bernstein, I think I mentioned. Justin Kinney's been trying to figure out how to get all the data analyzable, because this is a huge amount of stuff going back and forth. Leah Acker's been working on red light illumination specific strategies. I didn't have time to talk about Mike Henniger's work, but he's trying to figure out new kinds of camera probe that we could use to detect light. Scott Arfin is working on some of the electronics for these 3D probes. Steve Bates has been doing, along with Suhasa, the analysis of single cells. Mike Barada I mentioned. And Stephanie Chan was an undergraduate who worked on some of the early surgery automation work, which has now led to some other work they've been doing. Tanya Morimoto was an undergraduate who helped with the red chenoropsin finding. Alison Dobry was the undergraduate who started mutagenesis work on the ArchT class molecules many years ago. And Xiaofeng Chen and Mingxi Li and Amy Yang and a few other people have been helping with all the virology sides of things, which are, of course, part of the core of getting things done. So thank you.
So that was an incredibly lovely talk, and we'd like to give you this as a token, and then hopefully you'll take some questions.
OK, great. Yes. Yes.
Two quick questions. Do you think the problems in driving this with photon radiation that a decent contrast will ever be beaten? And also, where do you think the progress will go with coupling the channelrhodopsins to immediate early genes that have them driven by CFOS or?
Yeah. So there's a paper that just came out in Nature, I think today, from Susumu Tonegawa's group, where they made a FOS channelrhodopsin transgenic mouse. I haven't had time to even read it yet, but it sounds like the transgenic technology allows you to balance the gene dosage and some of the other issues where, if every cell has a different number of copies, then that will lead to some noise in the system. A transgenic where every cell has the same gene dosage seems like they were able to get reasonably good data using that. The first question on two-photon microscopy. So there's been a lot of innovation in the two-photon aspect. I think part of the problem is just that people are trying to hit a 7-nanometer thin membrane and scan, and so 99% of the volume is not useful for photoactivation. So several groups have come up with strategies that have resulted in improvements. So for example, in animals that can tolerate very high levels of expression, like zebrafish, just regular two-photon microscopy seems to have been good for getting two-photon activation. And several groups have been able to do that in zebrafish. So boosting expression, boosting current, I think, are important. But at least 3 or 4 groups now have also come up with good scanning patterns that allow you to focus on the membrane, either by shaping the light, using generalized phase contrast or spatial light modulation. Or one group even had just a very simple scan pattern to scan in a spiral, so that you can very efficiently and rapidly get to the membrane. But unlike a fluorophore, which has a nanosecond lifetime, right? These molecules can stay open for milliseconds. So even when you shine light on the membrane, most of the photons are wasted because the molecules are already open when you're in saturation. And so boosting the number of molecules would help. Increasing the open lifetime might help, but also hurt, because now you have less. The photons are not being used anymore there. I think there's a number of things that people are doing, but it's very species and cell type dependent, apparently, from talking to the different groups. Yes.
So, can you tell us some more about these really cool light probe technologies that you're working on?
Sure.
And so, how far, what kind of light, irradiance, are you getting out of those, out of those probes, and how far of a sort of, let's say, neuronal effect do you think that you're getting?
Right.
Or are you at that stage yet?
We're getting there. So the amount of light out seems to be limited by the micromirror device right now, because the mirrors don't tile it completely, and so light that doesn't hit the mirror is going to heat up the device. The waveguides have pretty good characteristics. So I can show you what they look like for some of the most recent batches. So this is width of an individual waveguide, and this is the loss in dB per centimeter. And so we have almost 0 losses at 10 to 20 microns. around 5 microns, it's still pretty good. And then you see this huge increase as you go from 4 microns to 3 microns. That's why we're mostly looking at 5 to 10 right now. But if we're out in the 5 to 10 range or even bigger, then the waveguide is not the limiting factor, because there will be very little heat losses in the waveguide. That said, we're kind of looking at 2 geometries. So for the most part, at this point in the sort of arc of neuroscience, stimulation, people want to stimulate small things, or at least they want to bother with this. And for silencing, people want to silence larger regions, because silencing one cell at a time is probably not yet a great experimental design, unless you work in C. elegans or something. So we're looking at 2 models right now. One model has these apertures being pretty small, 10 microns by 10 microns or smaller. And the other has them being larger, maybe 10 microns by 300 microns or bigger. And the former will be good for very sparse or localized photoactivation, and the latter for photoinhibition. That said, the amount of light that we can get out of, even with the DMD limits, is pretty good. So we're getting irradiance of around 150 to 200 milliwatts per millimeter squared out of these waveguides, even with the DMD limits that occur. And now we're thinking about, now that we know where the limits are, can we build laser arrays? Can we build a custom DMD, or get somebody else to do it, or buy one, or collaborate with somebody or something that would be an optimized system that would really enable these limits to be overcome. I mean, just imagine a DMD that had like some reflective coatings in between that would deflect the light some neutral position, and that would eliminate the heating issues. So some very simple designs might be enough to improve these kinds of things. And so, yeah, but that's where we're at now.
Can I follow on?
Mm-hmm.
So, I'm one of the few people in Cleveland that has been a big optogenetics and possibilities for clinical translation, supporter.
Uh-huh.
And so, but the monkey results that have come out so far haven't been super encouraging, probably related to a volume of activation issue. And so, so what do you think about the prospect of, as opposed to more focal stimulation, more diffuse, LED, kinds of array stimulation? And is that something that you think is realistic or not?
Good questions. Okay, so there are a couple of things. One is indeed how much of the brain can you sensitize to being virally or otherwise labeled. And that, I think, though, is a field that has its own driving forces. So, for example, new serotypes of adeno-associated viruses are being developed that can spread further. We just started playing around with the AAV-DJ variant, which was from, I think, Mark Kay's group, where they did some shuffling of the coat proteins and between different neuronally tropic. And we seem to get really, really good infection with that. Much bigger volumes than with even the sort of classical serotypes. So I think that's something where technology development trends are going to continue to push that, and already I think are quite good. I don't know of many publications that use DJ yet, but I think it's probably one of the better ones to try. And we're right now we can't get it made by companies, so we just make it ourselves with a kit. But you can buy the kit. Write to me if you want more information about that. The second thing is, of course, the light. So I think redder colors are going to be very important. I kind of skipped over the slide on what Leah's been doing with broad illuminators, but maybe it's worth mentioning because it's actually kind of a nice little hack. So red light, so if you use a very thin probe and you have red light coming out of it, and then you look at how the light goes, the light falls off very rapidly just because it's the 1 over r squared surface area of a sphere thing, right? As you go farther and farther away from a small aperture, you quickly have the light power falling down. So if you, I don't know, if a 200 micron fiber, so the surface area is what, 0.03 square millimeters, and then you look at a sphere that's 1 millimeter diameter, which is 3 square millimeters, that's 100-fold light power down, right? What if you have an illuminator that can illuminate in all directions, though, or an illuminator that has a broad illumination? Then the 1 over r squared effect goes away. It's replaced by a smaller power, or in the near range goes away entirely. And that's how you can get light maybe half a centimeter away or more. And that's easily done because if you have a very thin fiber, even though it has a lot of power, and you put it on and as red light you put it on your hand, it's going to scatter and it's going to look very dim. Take the same irradiance, have a wider thing, a wider illuminator, and even like a low-powered flashlight, right? You can see sometimes through your hand. So changing the geometry of the illuminator is also very important, and that's something that we've been doing to think about co-evolving kind of the illumination structures as well as the molecules to be optimally matched to each other.
So I could be wrong, but I remember with like the standard Michigan array, they had issues with scarring around. Do you know how that might impact like the opto characteristics? Have you kind of looked into that?
Yeah, so this is not my area of expertise. I think that 99% of the papers that have been published so far have just been focusing on short-term use of these for scientific experiments, lasting a couple weeks tops. And for example, the primate experiments tend to use acute probes. So the kind of detailed analysis of how these materials, glass and so on, are going to interact with the brain, I would say is sort of a new area. Yeah, so I think that'd be interesting to know more about, and whether the strategies of changing the the material stiffness or changing the coatings or changing the kinds of other strategies that are used, could they be applied to these kinds of things, or what are the optimal shapes and sizes? Very little's been studied with that. That said, the glass fibers can be quite flexible and quite thin, and also because it's a waveguide, right? It can be anything as long as you have two different refractive indices, right? You can make a waveguide out of many kinds of materials. So the hope is that this kind of thing might be fairly easy to evolve, because we don't necessarily need lots of onboard circuitry and silicon and the kinds of things that we might be more worried about if it was an electrical thing. But to be honest, I don't know.
Yeah, when you mentioned the channelrhodopsin 87,
Yes.
the one you developed, you were saying that the expression is faster than the older channelrhodopsin. So I was wondering how you get this auxin to express faster. Is an independent process of the promoter, or how it's possible that it's expressed faster? How you explain from the...
Well, the only thing different between this and what many groups have published before is the change in the opsin itself. The promoters are similar. We're electroporating because we wanted to avoid viral latencies where AAVs take a month to express and so on. We want it to be a really fair comparison of electroporating into the neural progenitors and then directly have them off to the races. So it could be a better folding protein for all I know. I mean, we're just starting to, we have this grant that should start, I think, in June, and we'd like to hire somebody to help us figure that out. But yeah, it's kind of a new area. What are the principles of heterologous expression of a protein in a foreign species, and what are the side effects that one has to look at, for example? I mean, a very simple example is in the native algal species. These are all localized to those little eye spots, right? And here they are covertly somehow being trafficked to the cell surface quite nicely. What signals are mediating that? Are there other channels being displaced for all we know, right? Those are questions that have not been asked yet.
Okay.
But I think they're important.
Okay, and another question is related with the red line, with the red-shifted channelrhodopsin. How far can you go? What is the penetration of the light? How far can you have the source of the light to the cells that are sensitive to light?
So, well, with the newer illuminators, this is all computational modeling, I should point out, but the data is coming in and it looks pretty good too. If we have, for example, a wide illuminator that's X millimeters wide, we get pretty good illumination X millimeters deep, because that's, we're not in the 1/r² regime, as it were. So that's kind of the rule of thumb that is 90%.
Is the possibility that just heat can activate the auxin?
Just heat can activate?
Yeah.
Oh, yeah, absolutely. I mean, so many groups using these tools for scientific purposes have noted artifactual neural activations, and then many groups that many groups actually are deliberately trying to use heat to activate neurons. I mean, there's several groups that have been been working on infrared neural stimulation, for example, and it looks very interesting. Yes.
As far as working towards the clinic and chronic long-term expression of this, how stable are these when they're in the viral delivery case? How stable with time are these, and what do you see as like kind of the big challenges that need to be overcome there?
Yeah, so a couple groups, including ours, have published studies looking at the duration of expression using AAV, for example. It seems to be, in mice, we do see expression out a year in, and that's as far as most groups have looked. One group has claimed to look at to almost 2 years, which is pretty much the limit, and they do see expression then as well. I do think some fall off does occur, which is kind of par for the course with AAV. But people who are doing AAV studies even in humans are doing these time courses, and I think consensus is that several years is feasible. Now, for the molecules themselves, do the molecules themselves have any changes over long periods of time? That also might depend also on the method of gene delivery and the gene itself. Any molecule expressed at a high enough level for a long enough time might be toxic, and you definitely do see toxicity with all of the molecules, even the original pteropsin 2. You express them at too high a level. At least reports, anecdotal or otherwise, have popped up about such things. So I think that one of the key things is that we're trying to do quantitative gene expression here, right? We want to express exactly the right amount. We want to express the right amount that allows us to do the actuation or silencing of the cell. The idea of just expressing as much as possible, which was how a lot of the early days of gene therapy was like, we just want to get as much as possible there. Now it's very much that you want enough to get X nanoamps of current, let's say, and more than that can only be bad, right? So we're doing a lot of things now trying to look at the interactions between promoters, gene dosages, and molecule types. And we're actually hiring hopefully a technician pretty soon to help us with all the histology. But there's some very, very subtle and nonlinear and even non-monotonic interactions where using a promoter that expresses at a very high level can lead to, we're being hand-wavy here, but maybe silencing of the gene expression, for example.